Présence. I'm Martha Crago. I'm the Vice Principal of Research and Innovation at McGill University. And c'est ma grand plaisir de vous souhaiter tous le bienvenue à ce symposium des chaires d'excellence de recherche canadiennes et les chaires 150 canadiennes. So welcome. While I regret that the pandemic is keeping us from holding the symposium on our McGill campus, I do look forward to the kinds of insights and networking that will emerge from this innovative virtual program. As we're meeting in a virtual environment, I'd like to acknowledge from coast to coast to coast of Canada, the ancestral and unceded territory of all First Nations, Inuit, and Métis people who call this land home. McGill University is on the land which has long served as a site of meetings and exchange among Indigenous people, including the Haudenosaunee and the Anishinaabe nations. And we acknowledge and thank the diverse Indigenous people whose presence marks this territory on which peoples of the world gather. The theme of the two-day symposium is Transformational Research for a Transformed World. And this is more than a simple catchphrase. It recognizes that the fight against the novel coronavirus has indeed transformed our world. In fact, we might say it turned it upside down. So during the last year and a half, researchers have been called on as never before to help overcome the many challenges that were presented by the pandemic. And what's more, it's put into stark relief the importance of national and international collaboration and cooperation. The CIRC and C150 programs were very ambitious and globally collaborative by design. In some ways, they anticipated the strategies needed to mobilize knowledge and maintain research programs under exceptional circumstances. So here at McGill, we're proud of the efforts we've made during the time of COVID. And one of these has been the rapid mobilization to house the directorate of the Biobanque Québécoise de la COVID-19. This is a $10 million initiative funded by the Public Health Agency of Canada, the Fonds de Recherche de Québec, and Genome Québec. And the Biobank is led by McGill's own Canada Research Excellence Chair in Genomic Medicine, Vincent Mousser. So this afternoon at 1:30, we'll have the opportunities to hear from Vincent and from 5 other CIRC and 150 chairs from across the country on their transformational research efforts during COVID-19. Upcoming panel discussions and presentations also invite us to look beyond the pandemic to the significant and socially relevant advances led by CIRCs and C150 chairs that are moving research and indeed societies toward more sustainable and equitable futures. So I am particularly looking forward to a conversation with Canada's research granting agencies presidents this afternoon, and it'll give them a time to reflect on the importance of this program, but also on research more generally in Canada. And then you, like I often do, can ask them questions about where you think the future of research in Canada is going. So if you haven't already looked at them, I invite you to go to the website in which videos were submitted by CIRCs and 150 chairs that highlight their research activities, and they'll also play during the break periods. So now I want to invite some distinguished guests to deliver their welcoming remarks. First, we are honored to share a message from Ali Hassasni, Parliamentary Secretary to the Minister of Innovation, Science, Industry. He's a member of Parliament for Willowdale, Ontario.
Good morning, everyone. It's a great pleasure to join you here today on behalf of the Government of Canada. In these changing times, I am heartened to be part of a government that understands the value of science and research. We recognize that we need facts to inform policy and program decisions. We know that evidence benefits our economy, our environment, our health, and the health of our communities. And we understand how basic science also leads to new innovations and solutions, which in turn lead to new markets, new skills, and the jobs of tomorrow. It is why, since 2016, this government has invested more than $13 billion in funding for Canadian researchers in science, including, of course, through the Canada Excellence Research Chairs and the Canada 150 Research Chairs program. Over the next 2 days, there will be plenty of opportunities for creating connections and new synergies. This is how innovation happens, and it could not be more inspiring during this period of international transformation. As a government, we are relying on you, the powerful minds behind the insights and solutions that will propel us into better tomorrows. And so, I wish you all the best for a productive and incredibly invigorating symposium. Thank you for all you do on behalf of Canadians and indeed all global citizens.
Well, thank you, Mr. Assasi. It's now my honor to invite my esteemed colleague, Ted Hewitt, President of the Social Sciences and Humanities Research Council and Chair of the CIRC and C150 steering committees, to say a few words. Ted, you're muted. Yeah. There we go.
Okay, I'm fine. I was hoping people could read lips because I was saying the host has to unmute me. Thanks, Martha, for your introduction, and I think most especially for your many valuable contributions to Canada's research and innovation ecosystem. Great to see everybody here today, this morning, this afternoon. It's just been far too long since we've had time to really spend some time together and to hear about the great work that you're doing. Je me fais l'écho des éloges des autres conférenciers à l'égard de l'équipe exceptionnelle qui a organisé cette activité. Et je sais que cela a été un défi en cette période de pandémie. Et je suis redevable à tous ceux qui ont travaillé si dur pour faire de cette rencontre un grand succès. Des conférenciers virtuels comme celle-ci auraient pu être considérés comme inconcevables en 2008 lors de la création des chaires d'excellence en recherche du Canada, ou même en 2017 lors du lancement des chaires de recherche Canada 150. While meeting virtually has undeniably been accelerated by the pandemic, it's really just one illustration of the rapid rate of transformation and adaptation to change taking place really on multiple fronts. From the development of safe and effective vaccines to fight COVID-19 in record time, to a helicopter flying remotely on Mars, to humans coining the term languishing as the emotion of 2021, there is no shortage of revolutionary developments that have pushed the boundaries of discovery. This reality reinforces the necessity of transformational research for a transformed world. It also underscores the crucial role that the CIRC program and Canada 150 chairs play in putting our country at the forefront in fields as diverse as artificial intelligence, cosmology, climate economics, and moral psychology. Nous sommes très fiers que des chercheurs de renommée mondiale aient choisi le Canada pour poursuivre de tels travaux. Et nous vous sommes reconnaissants du leadership dont vous faites preuve en encadrant et en formant des étudiants des cycles supérieurs et des boursiers postdoctoraux qui deviendront la prochaine génération de pionnières et de pionniers dans tous les domaines de notre société et dans notre économie. every reason to be optimistic about the future, as our summit agenda makes very clear. Presentations over the next days by CERC laureate and Nobel Prize recipient Dr. Michael Houghton, director of the Li Ka-shing Applied Virology Institute at the University of Alberta, and by Anil Arora, Canada's chief statistician, among other notable innovators, will undoubtedly leave us inspired with big takeaways. So with that, I'll draw my remarks to a close. I know we're all anxious to hear from our other speakers and to get on with the event. Bonne conférence, collègues.
Thank you, Ted. And now I welcome Danica Gusny, the Vice President for Research Grants and Scholarships Directorate of the National Sciences and Engineering Research Council, to say a few words, if somebody unmutes her.
I believe I am unmuted. So far, so good. Bonjour à tous et à toutes. Good afternoon, everyone. Merci, Professor Craigo, pour cette amiable présentation. C'est un honneur, bien sûr, pour moi d'être parmi vous aujourd'hui. I'd like to offer special thanks to Parliamentary Secretary Ali Assasi for his remarks moments ago. And I'd like to thank you, Dr. Craigo, and the McGill team for organizing such an incredible event to discuss transformational research for a transformed world. And oh my goodness, what a rapidly transformed world it is. The topics selected for this symposium cross disciplinary boundaries, just as the Canada Excellence Research Chairs and the Canada 150 Research Chairs programs do. Dans tous les domaines de recherche, il y a de grands défis à relever. Ces défis requièrent des objectifs ambitieux et un solide leadership. Combien de fois avons-nous vu des travaux de recherche transformateurs naître de la collaboration interdisciplinaire? Qui plus est, le changement porteur de transformation nécessite la plupart du temps une telle collaboration. Real-world problems can only be solved by creative, out-of-the-box thinking that emerge from such transformational research. Transformational research, of course, also requires excellence and leadership. As Canadians, we benefit in this regard in having not only a strong research community across this country, but also in particular, the constellation of research leaders supported by the CERC and C150 programs. Alors que nous faisons face aux défis que pose la pandémie de COVID-19, il est important de regarder au-delà de la situation actuelle et d'envisager les défis et les possibilités que se présenteront dans le monde après COVID. Les chercheurs et chercheuses dans toutes les disciplines auront un rôle important à jouer dans ce monde. One role is that of teachers and mentors. In order to sustain the entire breadth of the research enterprise for the future, we need to ensure that Canada not only has a strong scientific foundation for discovery and innovation, but that research capacity is built within the next generation of scientists and engineers. Nous devons aussi sensibiliser et inspirer les esprits innovants et les leaders d'opinion afin qu'ils puissent appliquer et communiquer ces nouvelles connaissances. Enfin, avant de céder la parole à Professeur Craigo, j'aimerais vous remercier tous et toutes de votre participation au symposium. Je suis impatiente de vous entendre et d'en apprendre de vous. Merci beaucoup. Bonne journée. Thank you very much.
And thank you, Danica. So now I'm pleased to welcome Michael Strong, the president of the Canadian Institutes of Health Research.
Merci, Martha. I'm assuming I'm unmuted. Terrific. So merci, Martha, pour votre participation aujourd'hui, for organizing this. And bonjour tout le monde with this. You know, I want to echo many of Ted's comments as well, in that this is such an honor to be able to welcome you to this symposium. Both to celebrate as the Canadian Excellent Research Chairs and the Canadian 150 researchers, but also to be able to take stock of where we are right now and where we really want to go as a national research ecosystem. It's an opportunity for us to say thank you to Canadian researchers from across the country who have really demonstrated remarkable perseverance throughout this pandemic. Really, from the moment that the WHO declared COVID-19 a global pandemic, Canadian researchers really have been at the forefront of helping to find solutions and to drive the research questions. You found, in a proactive manner, ways to contribute and collaborate more quickly, to submit research proposals as part of our rapid research response. And as a result of all of this, Canada has really emerged as a world leader in the global effort to generate the research evidence needed to address many of the challenges that we have faced and will continue to face as a global community. But this didn't happen overnight, and it didn't happen in a vacuum. To paraphrase others, it takes years to be an overnight success. And this really speaks to the foundational nature of these programs and of the research programs themselves, to really build the capacity and then the need to maintain this capacity in a steady state of knowledge creation. I think if we've ever seen a better example of this, I don't know what it would be, than our ability to respond during a pandemic for this. But it's not just on the knowledge creation side of the equation, it is equal as well on the knowledge mobilization. For as we've heard, and as we've heard from the Undersecretary, for the Secretary earlier today, this concept of being able to use science to help inform policy has never been as vibrant as it is now. And so this continuum of research from basic fundamental creation of new knowledge, which takes time and investment, through to translating that into policy to help inform our way out of a crisis, is really important. And so hence your theme of transformational research for a transformed world really does hit the mark, and really speaks to the needs of the Canadian research enterprise at this moment in our history. As we've all said, and we all continue to say, this last year and a half has been transformative and been unlike anything any of us have ever experienced for that. We've seen so many new realities, whether it be lockdowns or seemingly overnight development of vaccines, which we know were never overnight, but also the severity of what happens when labs are closed, what happens to that research ecosystem, and to the social upheaval as well. We see this in rising global calls for equity, justice, and an end to racism. So I look forward to the breadth of your discussions over these next 2 days, and for the contributions that you will continue to make to Canada's vibrant health research ecosystem. And I look forward to hearing other comments, and particularly to our discourse later in the course of today. So thank you. And again, I wish you tremendous success. Back to you, Martha.
Thanks a lot, Mike. And now, last but by no means least, please welcome Pierre Normand, who's the Vice President, External Relations and Communications of the Canada Foundation for Innovation.
Merci Martha. Bonjour à tous. Hello everyone. C'est un très, très grand plaisir d'être avec vous aujourd'hui au nom de la Fondation canadienne pour l'innovation à l'occasion de ce symposium sur les chaires d'excellence en recherche du Canada et des chaires de recherche Canada 150. We look forward to this exciting opportunity to celebrate the success of these pivotal programs in Canadian research. The Canada Foundation for Innovation was created almost 25 years ago to ensure researchers had access to state-of-the-art research facilities and tools they needed to think big and innovate. And today, campuses across the country are vibrant environments where researchers often are leads in their field. The outcomes of the research being done in the labs we've supported over the years are evident in the many benefits they provide to Canadians, from new technologies to a better understanding of our natural environment, to health solutions, and much more. If there is one thing we've learned in the past year, it's the need for cutting-edge research. You see, the COVID-19 pandemic clearly demonstrated that investments in research infrastructure and innovation are critical to our well-being. But of course, tools are not enough. We need the bright, ambitious researchers who will put them to use to create the new knowledge we need. And that's why the CFI created the John R. Evans Leaders Fund, and why, through this fund, we have partnered with the CERC and the Canada 150 Research Chairs programs. This partnership has made it possible for Canadian universities to attract and retain some of the best researchers in the world. To date, the CFI has invested $5 million to support the development of state-of-the-art research infrastructure and the work of 14 Canada 150 research chairs at 9 Canadian universities. As a result, these international leaders came to Canada to carry out their world-class research, and hubs of global scientific expertise were created in this country. We have also supported 26 Canada Excellence Research Chairs with over $20 million invested in the research infrastructure. In 2020, we did a study and found that for most researchers, the availability of infrastructure support plays a key role in their decision to come to Canada. You see, in the global race for talent, the CFI's support has made it possible to bolster Canada's place in research worldwide, helping Canadian institutions remain internationally competitive in areas of research and technology development that are aligned with their strategic priorities. The CERC and Canada 150 Research Shares programs also demonstrate that collaboration between the granting agencies can and does create the conditions and environment that serve as a magnet for global talent. The achievements we are celebrating today are the result of this collaboration. Et en terminant, j'aimerais dire merci. Merci à Dr. Craigo et à toute son équipe à McGill, à l'Université McGill, qui organise ce symposium, cet événement si important. And of course, I wish you all a very, very successful symposium. Thank you so much for your time. Have a great day, everyone. Bonne journée à tous.
Well, merci, Pierre. And let me second his statement about thanking the conference organizing committee. It's been a great job. We do have a packed program. So without further delay, I want to invite McGill's Dr. Luda Dyachenko, a CERC in human pain genetics, to introduce our keynote speaker, who is Dr. Michael Houghton, CERC and Nobel Laureate. And I wish everyone a productive and fun symposium.
Hello, everybody. So it's my privilege and it's my pleasure to introduce Michael Houghton. Michael was Canada Excellence Research Chair in Virology from 2010 to 2018. And I have privilege to overlap with Michael for 4 years in this capacity. And this was the time when we was getting together for summit every year. It was a great years, right? I see Steve was nodding, yes. So we should definitely continue to do this, because as everybody right now anticipating great interaction, this interaction was extended to personal exchange and just knowing each other. So, but it's great that we reconvene and welcome all the new chairs. It's great to see so many new faces. Well, I think we all was very happy to learn in 2020 that Michael was named 2020 Nobel Prize winner in physiology in medicine. I personally feel like this light shed over me too somehow. Yes, so the program really attract great people. So Michael, 2 words about Michael. So Michael was born in United Kingdom and graduated from University of East Anglia with Bachelor in Biological Sciences in 1972, and subsequently completed his PhD in biochemistry at King's College, University of London in 1977. Since that, he was known for his discovery of hepatitis C virus. And so he was working on the hepatitis C vaccine for the last 30 years in both US and Canada. And so without further ado, I would like to use time wisely and give it to Michael to tell us about this great journey.
Thank you very much, Luda, and thank you very much to the CERC program for inviting me to talk. It's been a fantastic pleasure and honor for me to be part of the CERC program. And I do remember many great meetings, very enjoyable meetings that we had for many years. So assuming everyone can hear me okay, I seem to be unmuted as far as I can tell. So I want to talk today about another pandemic besides COVID, and that is the hepatitis C virus pandemic, and how we are making real progress in controlling this pandemic. So there are up to 2 million new hepatitis C infections every year around the world. WHO estimate there's somewhere between 70 to 120 million carriers of the virus globally. Most of those people are not diagnosed, and unfortunately will slowly but surely develop serious liver disease unless they are diagnosed and then treated with antivirals. Hep C accounts for around 400,000 deaths every year globally. And in Canada, there are more than 10,000 new Hep C infections every year, according to the Public Health Agency. It is a blood-borne virus, unlike COVID, which is a respiratory virus. Hep C is transmitted through blood, direct blood transfer and contact. And so risk groups include people who inject drugs, PWIDs, healthcare workers in frequent contact with blood samples, men who have sex with other men, especially when multiple sex partners have been in the equation. And babies born to HCV positive mothers can also acquire the infection vertically. And then in addition, around the world, in developing countries with less infrastructure than other countries, nosocomial infection does occur through medical procedures, hospital procedures, and other types of medical interventions where lack of sterile equipment is used. So hepatitis C virus, the field was really born back in 1975 with this paper from the NIH in Bethesda. And this was after hepatitis A had been discovered by Steve Feinstein and Bob Purcell at the NIH, and after Baruch Blumberg had identified hepatitis B virus in the 60s. Tests were developed for both of those viruses, and in 1975, it was a great surprise to everyone in the field to learn that the large majority of post-transfusion hepatitis cases were not due to either hepatitis A or hepatitis B. And so the term non-A, non-B hepatitis was coined. It took a long time to actually discover the etiologic agent. We published our papers in Science in 1989, so it took 14 years to identify the causative virus. So I trained in England, as Luda said, and I was working on human interferons. And then I migrated with my wife and young baby son to California to join a biotech company called Chiron in 1982 that was set up by a couple of University of California professors, Bill Rutter and Ed Penhote. I was keen to continue my work on human interferons, but after a discussion with Dr. Dino Dina at Chiron, I decided to devote my own lab at Chiron to try to identify the causative agent of non-A, non-B hepatitis. I was a molecular biologist, and in some ways it was a strange decision for me to make because we had no molecular handle on the causative agent. We had, of course, human samples from the patients diagnosed with non-A, non-B. Many groups around the world had developed a chimpanzee transmission model. And so it was possible to get samples from the infected animals. but polymerase chain reaction had not yet been discovered, which makes me feel really ancient. Also, unlike Hep B and Hep A, no specific antigen or antibody had been defined for non-A, non-B. There was no cell culture system, which was the breakthrough for HIV at the Pasteur Institute in Paris. We had very limited sequencing ability in those days. And as I mentioned, in other words, there was no molecular handle available for a molecular biologist for me to get involved. So why did I get involved? I thought it was a very worthwhile exercise, and I thought there's not much else I could do that, if successful, would be more valuable. So as a molecular biologist, we tried many approaches. The main one was to try to identify messenger RNAs present in non-A, non-B infected liver samples that were unique to the infectious tissue. In other words, were not mRNAs that you could identify that were present in uninfected tissue. There are variations on that theme that you can try, and we did for many years. Found a lot of interesting genes that were upregulated and downregulated as a result of infection. But unfortunately, despite some interesting findings, we could not determine that any of them were of viral etiology. In those days, in the 80s, it was very confusing if you read about non-A, non-B hepatitis, because there were lots of reports that it was related to hepatitis B virus, toga viruses, picornaviruses, flaviviruses, retroviruses. So it was a very confusing field. We did try to use known viral genomes as hybridization probes to identify the causative agent without success. My lab took a detour to identify the genome of hepatitis delta virus, or HDV, because there was some evidence from the NIH that it might be a relative of Hep C. So it was a nice detour scientifically. We showed that hepatitis D virus is really a very basic, primitive, viroid-like element derived from plants, infectious agents of plants. So it was a really unusual element to show up in mammals, especially humans. But unfortunately, when we used it as a hybridization probe, it did not pick up hepatitis C. We now know hepatitis C is really a new, very novel virus. We tried to purify virus from infectious livers and blood. We tried to grow it in various cell lines. We assayed for reverse transcriptase activity. We tried to screen for specific antibodies and antigens, et cetera, et cetera. So we tried a lot of different approaches in the 7-year period up to our success. The successful approach is shown in this slide. We took chimpanzee plasma that was known to be infectious for other animals, and the original chimpanzee had been infected experimentally with a contaminated batch of human factor VIII, blood clotting factor. So we took this blood and we ultracentrifuged such that the smallest virus known to man would be pelleted. We didn't know if it was an RNA or a DNA virus, so we extracted total nucleic acid, converted it into DNA that we could then clone into an expression vector, lambda GT11, developed by Dr. Zhang and Davis at Stanford in 1983. And this is a very efficient cloning vector of nucleic acid, but it also expresses the nucleic acid into its encoded protein. And so we ended up with millions and millions of clones, each expressing the protein encoded by that particular cloned nucleic acid. So this was essentially an early proteomics approach. We then took serum from patients diagnosed by exclusion with non-A, non-B hepatitis, we assumed that there were antibodies circulating. And so we mixed the antisera with the proteomic libraries that were blotted onto nitrocellulose filters. And then we were looking for positive interactions. It was a very risky approach for various reasons, but eventually, after about a year and a half of trying this approach in different ways with different antisera and different materials as starting cloning materials, we found a small clone, 511, only 100 base pairs. And one of the first things we did is to find out, is it derived from the human genome or the chimpanzee genome? So this is a southern blot, another old technique that was in common use at the time. This lane are markers, molecular weight markers, and you'll notice that the rest of the blot, there's absolutely no signal. In contrast, over here is an old favorite gene of mine, the human interferon beta gene. It's a single copy gene, and you can see that we could clearly detect it in this analysis. And so we could not detect it with 511, and that told us that it was extra chromosomal. It was not a human or chimpanzee gene. We then went on to show that it was derived from RNA, isolated from infectious tissue. DNA was negative. The RNA present in the plasma as a virion was sensitive to ribonuclease, was resistant to deoxyribonuclease, confirming its RNA nature. Using strand-specific hybridization probes, only one of the strands hybridized, shown here. The other strand did not hybridize. And then on a northern blot, we showed that the 511 was derived from a very large RNA going up to around 10,000 nucleotides in length, typical of large viral RNA genomes. The next thing we did, and this was around 1987, 1988, we developed a Western blot for the 511 encoded protein. And then we looked for seroconversion in chimpanzees initially that were either infected experimentally with non-A, non-B hepatitis, or with hepatitis B, or with hepatitis A. And we looked at many chimpanzees infected with each agent. We only saw seroconversion from animals that had been experimentally infected with non-A, non-B. So this is day 0, day 76. There were still no antibodies, but 4 months and 5 months later, you started to see antibodies developing to the 511 antigen. And these were absent in Hep B and Hep A infected animals. Additional proof that we had actually identified Hep C came serologically. At this point, we were just using these GT11 blotted filters as the assay, and we received 11 clinically diagnosed non-A, non-B cases from Dr. Gitnick in UCLA, and we found around 60–70% positive for antibodies to 511, 0 out of 10 in controls. And as we was walking the genome and getting more and more sequence of clones overlapping with 511, we started to see very distant sequence identity with flaviviruses like dengue virus. And that was very reassuring to provide yet more confirmation that we really did have a new viral RNA genome. And so at the end of 87 approximately, we did believe 511 was derived from a non-A, non-B hepatitis genome. We then went on to look for antibodies to the proteins encoded by this virus in United States derived non-A, non-B cases. We worked with Dr. Harvey Alter at the NIH, a co-recipient of the Nobel Prize with me. And we were able to show in 9 out of 10 of his cases, 10 out of 10 of his cases were positive. And in 9 out of 10 of those cases, we could identify a positive blood donor with antibodies to proteins encoded by this virus. And then when we looked with other investigators shown here, we looked at another 24 cases of chronic post-transfusion non-A non-B hepatitis. We found that 71% were positive, yet in blood donors from the US, only 0.5% were positive. And then we worked with the CDC to identify positivity in 58% of cases of so-called sporadic non-A non-B hepatitis. These were cases that were not identified as a result of blood transfusion or blood exposure. And that's why they were called sporadic. We now know that most of those cases were in fact parenteral blood exposures. And then we started collaborating with other people around the world, with Drs. Bonino and Colombo. We found that 84% of their cases of non-A non-B were positive, 78% of cases from Japan, from Drs. Katayama and Miyamura. And interestingly, from those investigators, we got 13 cases of early acute non-A non-B infection. And we only found 15% were positive. So these antibodies, once the virus is eradicated, the antibodies do decline and become undetectable. So this work then, it seemed like an age at the time, but over 7 years, we were able to publish back-to-back papers in Science, saying that we had identified the causative agent of non-A non-B. And many colleagues were involved in this work with me, as you can see from the author list. I would especially like to acknowledge the contributions of Quilin Chu, George Kuo, and Dan Bradley. This was a team effort. It lasted a long time. You have to follow many different ideas, many different approaches, and you need dedicated, skilled collaborators to be successful. And that was the case here. Dr. Chu and Dr. Kuo were colleagues of mine at the Chiron Corporation, where I did this work, and were very, very important in the discovery. And Dr. Bradley was our collaborator at the Centers for Disease Control, who provided us with a first-class and a continuous supply of excellent chimpanzee samples throughout our work. So really acknowledge my colleagues in this success. So hepatitis C virus is now classified in its own genus, the Hep C virus genus, as part of the Flaviviridae family, along with flaviviruses like yellow fever, dengue, and also along with pestiviruses like hog cholera virus and bovine viral diarrhea virus, which are related but distantly to Hep C. And there's a group of viruses called the GBV viruses, which are viruses that infect marmosets, which also, as you can see, are distantly related to Hep C viruses. Excuse me. So eventually, by walking the genome, we identified the whole sequence of hepatitis C virus, a large RNA molecule of around 10,000 nucleotides in length. It encodes a single polyprotein from a single open reading frame. And the polyprotein is processed co-translationally as well as post-translationally into 3 virion structural proteins, the nuclear capsid, envelope glycoprotein 1 and 2, processed from the N-terminus of the polyprotein through the action of host signalase in the endoplasmic reticulum. And then a large bunch of non-structural proteins are cleaved from the polyprotein, which are involved in replication of the virus on the endoplasmic reticulum of the hepatocyte. So the first priority was to develop blood tests to prevent blood transmission from infected blood donors to recipients. In the 80s, in the United States, if you had a blood transfusion, the probability of leaving hospital with non-A, non-B hepatitis infection, which in the majority of cases is a persistent infection for life unless treated, the probability was in the region of 5, even sometimes as high as 10%. So a very big risk of getting a blood transfusion, which of course was saving the life of the recipient, but nonetheless, the byproduct was walking away with a slow virus that over many years, if left untreated, Dr. Alter showed would develop into liver cirrhosis, and Dr. Kiyosawa showed it would develop into liver cancer. So what we did is we identified all the various proteins encoded by the virus. We identified the major B-cell epitopes to which antibodies bound, and we developed a series of antibody detecting blood tests in ELISA formats. And this shows the estimated incidence of acute hepatitis C during the 80s and into the 90s. Our first test was introduced in 1990, and you can see that that resulted in a drop in the incidence of Hep C. Our second test was introduced in 1992. That introduced a further drop in acute Hep C. And all in all, we developed, I think, 5 sequentially more sensitive antibody tests for Hep C, as well as a nucleic acid test. The 5' untranslated region of the genome is highly conserved and actually shows some sequence conservation with toga viruses. And it's a perfect region to actually make PCR-based nucleic acid tests. And so now blood banks use antibody tests and nucleic acid tests. And thankfully, after experiencing a peak incidence of 5 to 10% after transfusion, post-transfusion Hep C has been eliminated. Of course, by this time, the field of hepatitis C virus was born, and we started to have regular international meetings beginning in 1992. And given the enormous number of carriers around the world who were replicating the virus in their livers, and during which, slowly but surely, the liver would be degraded and serious consequences would result in up to 20% of infected people, we had to develop new therapeutics. And of course, knowing the viral organization was a great help. NS5B was shown to encode the RNA-dependent RNA polymerase that replicated the genome. NS3, we and others showed encoded a chymotrypsin-like serine protease that cleaved in these positions here to release these individual non-structural NS proteins, all involved in replication of the virus. And NS5B, then the polymerase and this protease became very active targets by the pharmaceutical industry to develop small molecule inhibitors, which they did. A group at Bristol Myers Squibb developed the most potent antivirals ever made against any virus, which targeted NS5A protein. It's an unusual protein. It's not a classic enzyme by any means, but it is absolutely involved in replication and secretion of hepatitis C virus. And through some brilliant drug discovery and development work, declatosphere was developed as a very, very potent antiviral to HCV, as was suvosbuvir, an inhibitor of the replicase, and many inhibitors of the protease were developed over time. Well, it was surprising in some ways that it took so long to go from the discovery of the virus to very effective antivirals. It took actually 25 years of tremendous effort from academia and from the pharma industry. But eventually, in 2015, Jordan Feld, our own Jordan Feld, an outstanding hepatologist and virologist in Toronto at the Sick Children Hospital, as well as Graham Foster, published papers on the results of a phase 3 clinical trial using sofosbuvir, the polymerase inhibitor, along with velpatasvir, the NS5A inhibitor developed by Gilead. And after treatment for 12 weeks, you can see that irrespective of the genotype of HCV, there was almost 100% eradication of virus. This is actual eradicating the virus after just 3 months of treatment. So this was a huge breakthrough in treating the large number of carriers around the world. And then finally, to curb this pandemic, we need to develop a vaccine. The WHO estimate there are nearly 2 million infections around the world every year. So really, antivirals are very helpful in curbing the disease and to some extent curbing the transmission rate. But ultimately, we're going to need a vaccine to curtail this pandemic. And so I've been involved, as Luda mentioned, in vaccine development for many years, much longer than it took to identify the virus, which sometimes is embarrassing, but it's not an easy virus to make a vaccine to, obviously. And what we focused on historically, and while I've been at the University of Alberta in Canada for the last 10 years, we focused on the envelope glycoproteins E1 and E2 as a foundation for a vaccine. I should add, I don't have time to go into the details, but let me just state that the correlates of immunity against HCV infection are both, unsurprisingly, neutralizing antibodies to the virus, as well as CD4 positive and CD8 positive T cell responses. This has been shown by a number of groups around the world. And indeed, these are the correlates of immunity against COVID as well. And most, if not all, viral infections. You need a good humoral response as well as a cellular response. We made the envelope glycoproteins of Hep C, E1 and E2 in mammalian cell lines that we had transfected. And then we immunized chimpanzees while I was still in the US with various adjuvants. And then we challenged those animals experimentally with virus after vaccination. The chimpanzee model is immunocompetent, and it is the only reliable animal model for hepatitis C virus. Fortunately, the animals get infected, but they do not suffer disease from hepatitis C. And after their use in medical research, they are retired to sanctuaries. So what we were able to show is that E1, E2 prevented the development of chronic persistent infection in the animals. And to this day, this is the only vaccine candidate that has been able to do that. In the chimpanzee model, there are some other small animal models that are not totally immunocompetent, but there is no demonstration of prophylactic efficacy with any vaccine other than envelope glycoproteins E1 and E2. Highly heterogeneous molecules. This is E2, this is E1. They are highly glycosylated, which accounts for the large smear on acrylamide gels. Half of their mass is due to the carbohydrate, which acts as a shield against neutralizing antibodies, as in the case of HIV. And over the course of many years, using the chimpanzee model, using either homologous or heterologous viral challenges, we were able to show that we could significantly reduce the incidence of persistent chronic infection by prior vaccination. And you can see that of 31 animals that we immunized, only 5 went on to become persistently infected, meaning 16%, whereas of unimmunized control animals, 63% became chronically persistently infected with a highly significant p-factor, p-value. And so this is still the only vaccine shown to be prophylactic. Now, Hep C is more variable than HIV, which we all know is extremely heterogeneous. So there are multiple clades observed around the world. Genotype 1 is the most common around the world. And here is a slide showing that in Canada and the United States, genotype 1 is the most common. The orange is genotype 2 and the yellow is genotype 3. But it does vary. In South Africa, genotype 5 predominates in the blue sector. And in North Africa, genotype 4 is extremely common in Egypt and other countries. But genotypes 1A and 1B are the predominant genotypes around the world. So if we're going to make a global vaccine, we need to protect against most, if not all, of these different clades or genotypes. And so what we've been able to show, is that despite the high heterogeneity of Hep C, E1, E2 derived from a single viral genome is capable of eliciting broad cross-neutralizing antibodies in chimpanzees. We published this in 2011. It was a collaboration with the NIH, and they showed that we could very efficiently neutralize the infectivity of genotypes 1, 4, 5, and 6. This is a cell culture infectivity assay where antibodies were mixed with the virus prior to incubation with the cells. You can see that we very effectively inhibited infection of hepatocyte cell lines by genotypes 1, 3, 4, 5, 6, excuse me. Genotypes 2 and 3, although were not as well neutralized, were still significantly neutralized by polyclonal antisera from our immunized chimps, even though they only received E1, E2 from a single genotype. And in humans, when I was in the States, we ran multiple phase 1 clinical trials, including a couple with E1, E2, and we accessed that antisera from the NIH who sponsored the clinical trials. And then my colleague at the University of Alberta, John Law, showed that we could see evidence for broad cross-neutralization of the multiple global genotypes in humans receiving adjuvanted E1, E2. So the colors refer to the different genotypes seen around the world. This is inhibition of infectivity in cell culture by genotypes 1A, which predominates in Canada, 1B, which predominates around the world. And we were able to see evidence for broad cross-neutralization in many of these human volunteers that received E1, E2. We noticed that some of them, like this one here, individual number 1, we could neutralize genotypes, we could neutralize genotypes 1, 4, 5, 6 very well, not so well genotypes 2 and 3, which I think correlates with what I showed you earlier in the chimpanzee model. There we go. Okay, so envelope glycoprotein E2 was crystallized, and the structure was elucidated using X-ray diffraction by a group at the Scripps and also at Rutgers in 2013, 2014. then we started to understand where neutralizing monoclonal antibodies were binding. And as you can see, all of these reflect neutralizing antibodies, also cross-neutralizing. These monoclonals were identified by different groups around the world as highly cross-neutralizing the different genotypes. And so the epitopes were mapped to the N-terminus of E2, to the central region of E2, to the C-terminal, and very important ones, AR4A and AR5A, actually require the interaction of E1 and E2 in order to bind and to cross-neutralize. And so a student of mine, Jason Wong, actually was able to show that E1, E2 polyclonal antisera competes with each of these monoclonal antibodies, whether we immunize goats, which is this slide. I'll speed up because I'm running out of time. And also, Jason showed that when he looked at the phase 1 clinical trial volunteers who received adjuvanted E1, E2, we could see significant inhibition where the antisera was inhibiting the binding of 4 of the selected highly cross-neutralizing monoclonal antibodies. So what this told us is that although it's highly variable, there are many highly conserved epitopes, and when antibodies target those epitopes, they are capable of cross-neutralizing most of the clades in the genus of Hep C virus. So this was a very important finding. I think it was seminal in terms of trying to make a vaccine to Hep C. We know that antibodies are a correlate of immunity, but we also know T-cell responses are, as I mentioned earlier. And the way that we're dialing those into our vaccine, rather than choose large protein domains, which contain conserved T-cell epitopes, but also highly variant T-cell epitopes, and often in the case of the variant ones, they dominate the T-cell response. What we're doing is we're focusing the immune response on highly conserved CD4 and CD8 epitopes using synthetic peptides. And my colleague Amir Landi has identified 4 synthetic peptides encoding multiple CD4 epitopes, multiple CD8 epitopes, highly conserved amongst the genus. It's going to be very hard for the virus to mutate and escape from these conserved epitopes. And in the terms of CD8 MHC restriction, we've designed these peptides to have alleles present in more than 90% of the population. The other crucial ingredient to our vaccine is choice of adjuvant. I don't want to go into too much detail here because of shortage of time, but you're looking here at a FACS flow chart of CD4 cells and CD8 cells responding to various epitopes. This is actually an experiment we've done recently with COVID, the receptor binding domain, where we've immunized mice with adjuvanted receptor binding domain, and where we've used 5 different adjuvants. So in this context, E1, E2 is actually the control stimulatory peptides. And the reason I'm showing this to you is that adjuvants, if you choose the right adjuvant, for example, adjuvant 2, you can, with a recombinant subunit protein, prime very strong CD4 responses that secrete TNF alpha and gamma interferon, as well as substantial CD8 T-cell responses secreting the same cytokines. And as you can see, this adjuvant 2 is a lot better than adjuvant 3, 4, and 5. So choosing the right adjuvant is critical to a subunit protein vaccine. And that's what we're doing. And where are we in our development status with the Hep C vaccine? we've converted a lot of our researchers into GMP manufacturers that you need to produce the vaccine for clinical testing. We have a beautiful facility at the University of Alberta in the Alberta Cell Therapy Manufacturing Group, which I believe was sponsored by CFI. It's a state-of-art manufacturing facility for clinical trials. And our team of researchers are now manufacturers to produce a vaccine to go into phase 1 clinical testing. Our process, I think by industrial scale-up procedures, can easily provide vaccine for more than 1 million people every year, and that would just be from one site. I think multiple sites around the world would be very effective at curtailing the global pandemic. We've had delays of various types, but we're planning to do a phase 1 clinical trial in Edmonton and Germany next year using funding, very good funding from government of Alberta. And after assuming the phase 1 works, is safe and immunogenic, we would like to perform a phase 2 clinical efficacy trial in the US and Canada, enrolling around 600 people who inject drugs. That will take a few years to conduct. We are in the process of trying to procure the necessary funding for that, which will cost around $25 million. I believe this vaccine has a very good chance at significantly reducing the incidence of chronicity by Hep C. And it is the chronicity which causes the clinical sequelae of Hep C infection. It's not the early acute phase of infection, which is pathogenic. It's really the consequence of having the virus in the liver for many years. And I think this vaccine has a very good chance at lowering the national incidence of infection in Canada and around the world. Canada currently has around 12,000 new infections every year. 65% of those occur in people who inject drugs. And so I think delivering our vaccine initially to young injection drug users who are seronegative, but who have high risk, will, I think, have a good chance of dramatically lowering the national incidence. This would be highly cost effective because if we treat carriers over the next decade with the expensive but very effective antivirals, the healthcare economic burden will be around $1 billion over a decade, whereas the cost of our vaccine would be in the region of just 30 million. So this is highly cost effective. And I believe that's a strong argument for trying to procure the 25 million funding for an efficacy trial. And certainly we would appreciate funding from the various funding agencies to help us do this once we have shown phase 1 safety and immunogenicity. Thank you very much. It remains for me to thank a variety of University of Alberta colleagues working with me, originally in my CERC lab until 2018, and then subsequently in the Li Ka-shing Applied Virology Institute that we set up in 2013. I'm indebted to Dr. John Law, Mike Logan, and Amir Landy, who are senior RAs in the lab, who are responsible for much of the work I've shown you. Have a fantastic lab manager in Darren Hockman. We've had 3 outstanding students who have really contributed to our understanding of the Hep C vaccine, Jason, Janelle, and Catalina, and then many other highly skilled scientists at the University of Alberta who have made significant contributions. Finally, my colleague, collaborator, and dear friend, Dr. Lorne Tyrell, who I've worked with closely in Hep C and our other activities while at the University of Alberta. I would like to thank Canada Excellence in Research Chair Program for the funding you gave me. The government of Alberta has been a strong supporter of our Applied Virology Institute. And by the way, we're about to start a phase 1 clinical trial of a group A streptococcus vaccine. which will be conducted in Edmonton. We have new cytomegalovirus antivirals that we're very excited about. And we're also working on novel approaches to Alzheimer's and the new hepatology challenge, which is non-alcoholic fatty liver disease. We have initial drugs inhibiting that process. Li Ka-shing Foundation has been really supportive of us. Alberta Innovates, Western Economic Development, and CIHR. I thank you all for the funding. And especially thank you to CIRC. It's been an honor and a great pleasure to be one of your chairs. I was, and still am, tremendously excited about the program in general, the wonderful scientists that are part of it. And I am sure, I'm totally sure, the benefits to Canada will emerge and already have, hopefully. And I really commend the instigators of the CIRC program, and thank you for my funding. Thank you very much.
Michael, thank you so much. So it was a great presentation. So it was a great story, and I think it should inspire all of us, not go for the low-hanging fruit, but take the, tough project instead of, high risk, high reward, and be persistent. And it's interesting to learn that the last part, which is developing vaccine, was the most difficult, was the lengthiest one. So unfortunately, we don't have time for the questions to keep the time. But I don't know, maybe I right now revealing some biggest secret of Michael, but I know Michael answer emails. So everybody, I would encourage you, if you have question, would like to follow up. And we have another 2 days, and so we have a chart. So please post your question there. And thanks again to everybody, and Michael especially. Thank you.
Thank you, Luda. I'm sorry I talked too long. Apologize.
No, it was delayed in the beginning too. So no, so it's not you. So I think.